Do we need new or safer rapalogs for aging prevention? The debate about whether new or safer versions of rapalogs, rapamycin and its analogs, are required for aging prevention continues, but the evidence suggests that the current formulations are already safe and effective for human use. Despite concerns about rapamycin's metabolic side effects, particularly in some animal studies, it remains a drug with proven benefits for extending lifespan and improving health span in a wide range of species, including humans. So, do we really need to develop safer rapalogs to treat aging, or are the current options sufficient? Rapamycin, a proven track record of safety. First and foremost, Rapamycin and everolimus are FDA-approved drugs that have been in clinical use for over 2 decades. Since 1999, these drugs have been administered to millions of patients, primarily for preventing organ transplant rejection and treating certain cancers, with no unexpected or catastrophic side effects. The metabolic effects seen in some mouse models, such as hyperglycemia, have raised concerns, but these effects have not been significant enough to halt its use in humans. In fact, elderly people who have been treated chronically with rapamycin or other mTOR inhibitors have not reported major issues such as hyperglycemia, suggesting that these drugs are well tolerated in human patients. Some researchers argue that while rapamycin is safe for sick patients, it may pose risks for healthy individuals. However, this argument misses a crucial point. By the time we reach old age, none of us are truly healthy in the sense that we are all susceptible to age-related diseases. The concept of healthy aging is somewhat misleading because aging itself is the accumulation of diseases. As such, the case for new rapalogs, not about safety, but efficacy. The need to develop new rapalogs is less about safety and more about further improving their ability to extend lifespan. Rapamycin and everolimus are effective, but their impact on lifespan is still limited. The goal of future research should be to develop drugs that can enhance the therapeutic benefits of rapamycin by targeting additional mechanisms beyond the ones already addressed by rapamycin and its analogs. Current rapalogs work by inhibiting the mTORC1 pathway, which reduces cell proliferation and leads to various beneficial effects on aging. However, mTORC1 also has rapamycin-insensitive functions that are not affected by current treatments. The development of new rapalogs that can inhibit these functions might unlock even greater anti-aging benefits. Importantly, the side effects of rapalogs, such as reductions in blood cell counts and insulin levels, are largely dose-dependent. Higher doses of rapamycin over a long period can inhibit cell proliferation, leading to these side effects. But these effects are reversible and can be managed by adjusting the dosage. Some researchers suggest developing weaker rapalogs with fewer side effects. But this raises a key question: why not simply reduce the dose of rapamycin? A lower dose of rapamycin would achieve the same reduction in side effects without diminishing its therapeutic benefits, which is why some experts argue that the push for safer rapalogs might be unnecessary for now. Current rapalogs, safe enough for now. Given that the metabolic side effects of rapamycin are well understood and manageable, the focus should be on making the best use of the rapalogs that are already available. As of now, there is no pressing need to halt or delay the use of rapamycin while waiting for new and safer alternatives to be developed. The current versions of rapamycin and everolimus are safe for long-term use in humans when administered correctly, and their benefits for extending lifespan and improving health are significant. Moreover, rapamycin is a natural compound, derived from a soil bacterium found on Easter Island, and its patent has expired, making it widely available and affordable. Pharmaceutical companies have already developed analogs like everolimus, which have a similar therapeutic profile. These analogs show the same side effects as rapamycin and, at the same dosage, provide comparable benefits. Conclusion: use what we have while we wait for the future. The call for safer rapalogs stems not from concerns about the safety of rapamycin, but from the desire to extend lifespan even further. While it is crucial to continue research into new analogs that may improve the efficacy of mTOR inhibition, the current drugs are already safe and effective enough to be used now. We should not delay their use in the hope of finding a silver bullet drug in the future. As science continues to advance, we may discover new rapalogs that work in conjunction with rapamycin or inhibit additional pathways. However, in the meantime, rapamycin and everolimus remain powerful tools for extending life and promoting health in older adults. Rather than waiting for the next breakthrough, the focus should be on how we can optimize the use of current rapalogs for aging prevention today. In summary, while new rapalogs may one day offer even greater benefits, rapamycin is already safe enough to be used for aging prevention in humans. The key now is to continue refining its use and exploring how it can be combined with other interventions to further enhance its lifespan extending potential.